This is the video of photosynthesis part 2. First of all, we should have a general idea about the various metabolisms that occur in plants. First of all, to speak, metabolism can be classified into photosynthesis, respiration. Photosynthesis again classified into light reaction through which oxygen is evolved, and dark reaction in which carbohydrate is formed. Respiration, the steps of respiration is glycolysis, which is anaerobic in nature, followed by Krebs cycle, followed by electron transport chain. This light reaction can be again classified into cyclic photophosphorylation, non-cyclic photophosphorylation. Okay, so there are various types of dark reactions like C3 Calvin, C4 Hatchlag pathway, C2 photorespiration, and CAM pathway. Okay, so this is a general outline about the various metabolism that occurs in the plant. First of all, we are going to study in this chapter about the photosynthesis. So where does the photosynthesis take place? Photosynthesis does take place in green leaves where chloroplast will be present, and it also does take place in the other green parts of the plant like stem. So within the chloroplast there is a membranous system that contains grana. This is the whole grana. Grana is made up of thylakoids. These are thylakoids, and these are stroma lamella, and the matrix stroma. So matrix stroma. So this stroma lamella has only one photosystem 1 is only present in stroma lamella, whereas grana has both photosystem 2 and photosystem 1. This photosystem are named according to the time of discovery. Photosystem 1 was discovered first. After that only photosystem 2 was discovered, but actually to speak, electron will get excited from initially from photosystem 2 only, then only electron from the photosystem 1 will be excited. So within the chloroplast there is a membranous system consisting of grana, the stroma lamella, and the matrix stroma. Grana, stroma lamella, and matrix stroma. There is a clear division of labor within the chloroplast. So the membrane system is responsible for trapping the light energy, so that is the photophosphorylation, and also for the synthesis of ATP and NADPH. In stroma, so I will come to that. In stroma, the enzymatic reactions synthesize sugar, that is stroma is where dark reaction will take place, and this grana and stroma lamella is where light reaction will take place. So enzymatic reaction synthesizing sugar where stroma, so which in turn forms starch, that is the polysaccharide that will travel to the phloem through the phloem. The former set of reactions, since they are directly light driven, are called light reaction, that is photophosphorylation, and not non-photophosphorylation, that is the photochemical reaction. And the later is not directly light driven, but are dependent on the products of light reaction like ATP and NADPH. Hence, to distinguish it from the light reaction, they are called as dark reaction. It does not mean it will occur only during the night times. It will occur both in daytime and night time, but the actual requirement for the dark reaction is ATP and NADPH, that is synthesized as a result of light reaction. From the light reaction only oxygen and ATP NADPH are evolved, and in dark reaction carbon dioxide is reduced to form glucose. Okay, let us see this picture. This picture shows a good a good this picture shows a good representation of photosynthesis. So from sunlight is coming, and this sunlight is exciting electron, and here light reaction take place inside the grana, that is thylakoids, pile of thylakoids and stroma lamella, where water is oxidized to give H plus and electrons. These electrons will be used for refilling of electrons in photosystem 2, and this H plus will be used to reduce the NADPH. Okay, NADPH or NADP. Okay, so as a result of this light reaction, ATP and NADPH are formed, and this ATP and NADPH will be involved in the formation of glucose in the dark reaction that take place in the stroma, thylakoid stroma. At last, what we need to know is both light reaction and dark reaction take place inside the chloroplast only, but this light reaction take place in the stroma lamella and thylakoids, but the dark reaction take place in the stroma matrix, because matrix has all the enzymes required for the synthesis of sugar. Usually these chloroplasts align themselves, that is along the walls of the mesophyll cells, okay, such that they get optimum quantity of incident light. So usually chloroplasts align themselves along the walls of mesophyll cells such that they get the optimum quantity of incident light. So in which conditions how the chloroplasts are aligned? High light intensity. During high light intensity, that is during midday, that is 1 p.m. or 2 p.m., 12 to 2 p.m., parallel to the incident light, lateral walls. The chloroplast will be aligned on the lateral walls, and low light intensity perpendicular to the incident light. So that is during the evening and early morning, and moderate light intensity that will be around 10 or 9 a.m. that will be random. Okay, so this is the various alignments, pictorial representation of alignment of chloroplast. So this is during the high light intensity, that is midday, 1 p.m. or 12 p.m. like that. So when the light intensity is very low, it will be aligned perpendicular to the light incident. The incident light is in this direction, means it will be aligned in this direction. So it is perpendicular to the incident light. But whenever the light is of high intensity, it will be aligned parallel to the direction of light. And when moderate light is present, the chloroplast will be aligned randomly. So the question they gave here is, when do you think the chloroplast will be aligned with their flat surface parallel to the walls? When under high intensity condition, that is called as parastrophe. And when do you think chloroplast will be perpendicular to the incident light? Under low intensity light conditions, this is epistrophe. So there are here comes the question, how many pigments are involved in the photosynthesis? There are 4 major pigments. Okay, these photosynthetic pigments are special molecules. Those absorb, transmit and reflect different colors of light from visible spectrum of sunlight. That is from 400 nanometer to 700 nanometer is the visible spectrum of light. So these various chlorophyll pigments absorb light in the range of 400 to 700 nanometer. Pigments appear in the color which reflects and uses the color which it absorbs. So these photosynthetic pigments are of following types. So chlorophylls, chlorophyll A, chlorophyll B, and carotenoids, some xanthophylls and phycobilins. Okay, so these are the various pigments involved in the process of photosynthesis. Bacteria like prokaryotes do not have chloroplast, but they also do photosynthesis with the help of these pigments. These pigments will be present in the cytoplasm itself, and that will do photosynthesis. And so these leaf pigments can be separated through paper chromatography technique, and the result of the separation is called chromatogram. So chlorophylls, we are going to study about the chlorophylls. So green color pigment, because this chlorophyll pigments absorb all other lights except chlorophyll. The green color, it will absorb all the spectrum except the green color spectrum. So light is required for their synthesis. Soluble in organic solvents. Chlorophyll is soluble in organic solvents. Chlorophyll can be of various types. Chlorophyll A and B are very, very important. Chlorophyll A is the universal pigment, that is the central part of the photosystem. You know, right, there are photosystems present, photosystem 1 and photosystem 2. So this chlorophyll A is a single molecule that will be present at the center of the photosystem, from which electrons will be excited. The remaining chlorophyll B, xanthophyll, carotenoids, they all will gather the energy, light energy, and they will transfer this energy to chlorophyll A, and from chlorophyll A is where from electron are excited. So this chlorophyll A is a universal pigment which is found in all oxygen liberating photosynthetic organisms. Its color is blue green in chromatogram. Chlorophyll B is an accessory photosynthetic pigment, and it is found in euglenoids, green algae, and higher plants. Its color is yellow green in chromatogram. So this is a general structure of chlorophyll, and it looks like a tadpole. And this chlorophyll has a porphyrin head and a petal tail. This chlorophyll A, it is having methane group in second position, and chlorophyll B has aldehyde group, methyl group in second position, and aldehyde group in second position of pyrrole ring. Okay, so this is the difference between chlorophyll A and B. Chlorophyll synthesis, this is synthesized with the help of succinyl CoA and glycine, and this reaction is catalyzed by iron. Okay, this reaction is catalyzed by iron. So these are the general important things. So carotenoids, coming to carotenoids, first carotenoid was discovered in carrot. It will be, the carotenoids are more or less yellowish orange in color, and these carotenes are the first type of carotenoids. These are yellow orange in color. Here I told you, right? So they are yellow orange in color, and it contains carbon and hydrogen. Another carotenoid called xanthophyll, that contains carbon, hydrogen, and oxygen. These are usually yellow in color. Some plants with yellow leaves, if you take those yellow leaves, will have xanthophyll. And among carotenoids, beta carotene, that is the active form of vitamin A, and lutein are commonly present in plants. Carotenoids, they are accessory pigments and make photosynthesis more efficient by absorbing different wavelength of light. They protect chlorophyll A from photooxidation and also protect photosynthetic machinery by converting lethal nascent oxygen, that is nascent oxygen means single atomic oxygen, to useful molecular oxygen O2, and thus called shield pigment. So which pigment is called as shield pigment? Carotenoids. Carotenoids are called as shield pigments. Coming to the final pigment, that is phycobilins. So they are hot water soluble pigments. They lack magnesium and phytol tail. So they occur exclusively. They occur exclusively in blue-green algae. BG is blue-green algae. That is the cyanobacteria. Cyanobacteria. It is not a algae. Blue-green algae is not an algae. It is a bacteria. Initially, it was misconception. It was a misconception that it is a algae, but later it was discovered that blue green algae is not an algae, it is not a eukaryote, it is a prokaryote, and it is a bacteria, the bacteria that does photosynthesis. Okay, so these phycobilins are exclusively seen in blue green algae, that is cyanobacteria, Monera, and red algae is nothing but an algae that is a class of algae present in plant kingdom. Okay, these phycobilins are acting as an accessory pigment. So absorption spectrum, so if we see the graph, so this is the absorption of light, how much lights are absorbed, and these are the various wavelengths from 400 to 700 nanometer. Okay, so photosynthesis is highly seen at this place and this somewhat here. Okay, right? So this is the photosynthesis, and these are the pigments absorption of light, which pigment is absorbing much light. Okay, so if we see chlorophyll A absorption and photosynthesis, they are correlated. So with the light, maximum light absorbed by chlorophyll A, maximum photosynthesis occurs. So this graphical presentation of the absorption of different wavelength of light by a particular pigment is shown here, and this figure shows that chlorophyll A shows maximum absorption at blue light. This is blue light, and it shows another absorption at red light. So whenever we grow a plant, if we use blue light or red light, then the photosynthesis rate will be high, then the productivity of that plant will be high. We need to use either blue or red light where photosynthesis will be maximum. It means the absorption of light by chlorophyll A is maximum at blue and red light. Okay, so the graphic curve depicting the relative rates of photosynthesis at different wavelengths of light is called action spectrum. So this is the action spectrum. So here from this what we understand is the wavelength at which there is maximum absorption of chlorophyll A, that is the blue and red regions, also show higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. This is very important. So from this graph we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. So now considering the interesting experiment done by T.W. Engelmann. So using a prism, he has split the light into its spectral components, right? If we use this prism, it will split the white light into various colors. The seven colors. Okay. So using a prism, he split lights into its spectral components and then illuminated a green algae, that is Cladopora. He illuminated the light on a green algae that they named Cladopora, placed in a suspension of aerobic bacteria. This Cladopora is placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. So this bacteria will detect the site where O2 evolution is maximum. So he observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. So a first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectrum of chlorophyll A and B. From this graph also you can see that at blue light chlorophyll B and chlorophyll A is having somewhat peak, and at red light also chlorophyll A and chlorophyll B are having a small peak. So this blue light and red light is where chlorophyll A and B absorbs maximum light, and these graphs together show that most of the photosynthesis take place in blue and red regions of the spectrum, At the same time, some photosynthesis does take place at the other wavelengths of the visible spectrum. It happens because other thylakoid pigments like chlorophyll, xanthophyll and carotenoids, which are called accessory pigments, they also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wide range of wavelength of incoming light to be utilized for synthesis, but also protect chlorophyll A from photo oxidation. This is very important. Why accessory pigments are present? What is the function of accessory pigment? If they ask, the answer is to prevent photo oxidation is the first answer. Second answer is it will help in the absorption of light at various wavelengths. Like these carotenoids and phycobilins can absorb light in different wavelengths, so because of their different color. So from this chapter what we understood is light energy is converted into chemical energy by photosynthesis. Photosynthesis is a redox reaction and it is an anabolic reaction. It is the structure forming reaction and endergonic, so it absorbs energy. Endergonic reaction, anabolic reaction, and it is a redox reaction. So redox reaction means one has to be reduced, one has to be oxidized. Water is oxidized and carbon dioxide is reduced. Water is oxidized into oxygen and H plus ions and electrons. Carbon dioxide is reduced to glucose, and the oxygen evolved as a result of photosynthesis is only from water and not from carbon dioxide. So there is a clear division of labor within the chloroplast. The membranous system, that is the grana, thylakoids and stroma lamella, that is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. And in the stroma, enzymatic reactions synthesize sugar like glucose, which in turn forms starch, and this starch is transported through phloem in a multidirectional way for the storage and utilization of this starch. And these leaf pigments can be separated through paper chromatography technique, and the result of this separation is called chromatogram. Coming to the next thing, the chlorophyll A is a universal pigment which is found in all O2 liberating photosynthetic organism. Its color is blue green in chromatogram. Phycobilins are hot water soluble pigments, and chlorophyll A shows maximum absorption at blue light. It shows another absorption peak at red light. So what is light reaction? Light reaction or photochemical phase includes light absorption, water splitting, that is chemiosmosis, oxygen release, and formation of high energy chemical intermediates like ATP and NADPH. So photosystems are required for this process. Photosystems are required for this process. Photosystems are nothing but photosystems are nothing but arranged pigments, arranged photosynthetic pigments. So these photosynthetic pigments like chlorophyll A, B, carotenoids, xanthophylls are arranged in a way so that they can excite the electrons from the chlorophyll A. Chlorophyll A will be present at the center of the photosystem. Photosystem is nothing but unique arrangement of these photosynthetic pigments to excite the electron from the chlorophyll A. If it excites electron, then only this photosystem can that excited electron can form ATP and NADPH. Okay, so this Emerson and Android, two scientists worked on chlorella and have gave concepts of two photosystems, photosystem 1 and 2 pigment systems. So Emerson effect and red drop. So what is it? What is called as Emerson effect and red drop? Emerson Android experiment upon chlorella. The observation is reported minimum photosynthesis yield when supplied monochromatic beam of light of 700 nanometer or 680 greater than 680 nanometer only. So that is beyond the visible spectrum. More or less they are reaching the end of the visible spectrum. So that is the reason why photosynthesis is reduced. Report enhanced in photosynthetic yield when both 700 nanometer and 680 nanometer supplied together. Enhancement effect or Emerson effect. So from this, what is the conclusion? Two types of photosystems. Photosystem 1 and 2 exist in photosynthetic units. They operate simultaneously and their operation activation requires 700 nanometer and 680 nanometer radiation. And the group of photosynthetic pigments in thylakoid membrane. This is what I am telling you. So the group of photosynthetic pigments in thylakoid membrane are known as photosystems. So what are photosystems? They are nothing but group of photosynthetic pigments in the thylakoid membrane is called as photosystems. The pigments are organized in two discrete photochemical light harvesting complexes. So LHC light harvesting complexes within the photosystem 1 and photosystem 2. So the photosystem has a reacting center which is nothing but chlorophyll A and light harvesting complex that includes chlorophyll B, xanthophylls, carotenoids, phycobilins. So this reaction center is chlorophyll A, from where electron is going to get excited. Surrounded by it is the light harvesting complex. They are nothing but chlorophyll B, xanthophylls and carotenoids and other pigments like phycobilins. So in every photosystem there is a reaction center molecule, a single molecule of chlorophyll A surrounded by accessory pigments, that is chlorophyll B, xanthophylls and carotenoids. So these accessory pigments absorb light energy and transfer it to the reaction center, that is chlorophyll A. These pigments help to make photosynthesis more efficient by absorbing different wavelength of light. So this is what I am also telling. So these molecules are known as antenna molecules or light harvesting complex. So photosystem has central reaction center and light harvesting complex. This light harvesting complex is otherwise called as antenna molecules, and these antenna molecules or LHC are made up of hundreds of pigment molecules bound to protein. So this is the grana. Grana has thylakoids. So if you see the membrane of thylakoid, it has photosystem 1 and 2 arranged. So this is the thylakoid membrane and it has photosystem 1 and 2. This photosystem has a central reaction center. Central reaction center is present. Okay, surrounded by it are protein bound chlorophyll pigments, that is light harvesting complex or antenna molecules that will help in the photosynthesis light reaction. So the reaction center is different in both photosystems. Photosystem 1, in photosystem 1 the reaction center, that is a chlorophyll A, absorbs a peak at 700 nanometer. Hence it is called P70. Photosystem 1 is P70, while photosystem 2 will have a absorption peak at 680 nanometer. So it is called P680. P700 means photosystem 1 and P680 means photosystem 2, because the absorption spectrum varies for both these photosystems. These are named in the sequence of the discovery and not in the sequence in which they function during the light reaction. And this photosystem is located in the oppressed region of granule thylakoids and photosystem 1 in non-oppressed region of grana and stroma thylakoids. In other way, we can say that the granule thylakoids have both photosystem 1 and 2, whereas stromal thylakoids have only one. That is, this stroma lamella has only photosystem 1. It means it can absorb light only at the wavelength of 700 nanometer, because it has only photosystem 1, and this stroma lamella is where cyclic photophosphorylation occurs. because excited electron cannot be replaced, so the excited electron will come back and sit at its initial position. So the electron is cycling, so cyclic photophosphorylation. But this grana has thylakoid membranes which has both the photosystems. It is where the non-cyclic photophosphorylation or the Z scheme occurs. So in this picture we can see, so this is the non-oppressed part. Non-oppressed part has photosystem 1 and the oppressed part has photosystem 2. Okay, these granule thylakoids have photosystem 1 and 2 and ferridoxin NADP reductase enzyme, whereas the stromal thylakoid has only photosystem 1. So if photosystem 1 is only present means then cyclic photophosphorylation will occur. If both the photosystem are present, then non-cyclic photophosphorylation will occur. So first of all we are going to study about the non-cyclic photophosphorylation that is taking place in the thylakoid membrane of grana, because it has two photosystems, photosystem 2 and 1. Initially electron from photosystem 2 will be excited as a result of the energy taken from the sunlight. So these electrons will get excited and they will be transported to photosystem 1 with the help of electron transport system. During the transportation, the energy from the electron is utilized for the formation of ATP, and this electron will go here and it will sit in the place which is evacuated by the excited electron from photosystem 1. Photosystem 1 will also excite electron at an wavelength of 700 nanometer. Photosystem 2 will excite electron at 10 nanometer at a wavelength of 680 nanometer. Okay, so electron will be excited from photosystem 1, so that empty space is occupied, empty space of the excited electron is filled by the electron that is coming from photosystem 1. So this photosystem 2 has, now it has empty space, that means it has empty space from which electron has, it has created an space initially where electron is present. Okay, so that electron gap should be refilled. That electron gap will be refilled with the help of chemiosmosis, that is oxidation of water will release H plus and electrons and oxygen. Those electrons will go and sit at the place where electrons are removed, from where electrons are removed, and the electron that is excited from photosystem 1 will form NADPH. These NADPH and ATP will be utilized in the formation of sugar during the dark reaction. So this depicts the chemiosmosis. Chemiosmosis is a process through which the empty space that is created due to the excitation of electron can be filled. So what happens is, with the sunlight, what happens, photosystem 2, from photosystem 2 electrons are excited, so there is an electron gap occurs. So to fill that gap, water will undergo oxidation to form 2 hydrogen and half O2 molecule, and 2 electrons will be formed. That 2 electrons will enter into photosystem and fill the gap created as a result of excitation of electron, and this H plus will be utilized. This H plus will be utilized for the formation of NADPH, or otherwise it can be used for some other various functions like reduction of carbohydrate also. Reduction of carbon dioxide to carbohydrate, and these 2 of O2 molecules will combine to form one O2 molecule that is liberated out as an oxygen. So this oxygen is evolved during light reaction only. So that is the reason why when people if they sleep under tree during night, what happens is during night there is no sunlight, so light reaction, there is no way for the light reaction to occur, so no light reaction occurs. If there is no light reaction means no oxygen can be evolved, oxygen is not evolved. At the same time plants are also living organisms, they will also do respiration, so as a result what happens is this respiration will evolve O2 and the plant cannot absorb O2 or it cannot evolve. During as a result of respiration, what happens is the plants will evolve carbon dioxide, that is CO2. That CO2 cannot be reabsorbed by the plant because it is not doing photosynthesis right now, so that CO2 will accumulate. At the same time the plant cannot evolve O2 because there is no light for the light reaction to occur, because light reaction, oxygen is evolved only with the help of light reaction. So both photosystem 1 and 2 are involved in non-cyclic photophosphorylation, so it occurs at the grana thylakoids only and not in the stromal thylakoids, because stromal thylakoid lack photosystem 2 as well as NADP reductase enzyme. Coming to the primary electron acceptors. So the primary electron acceptors of photosystem 2 is phaeophytin, which passes electron to the electron transport chain. So here if you see the electron acceptor here is it is present, right? So the primary electron acceptor of photosystem 2 is phaeophytin, which will transfer that electron to the electron transport system. So it is transferring the electron to the electron transport system consisting cytochrome. This movement of electron is downhill in terms of an oxidation reduction or redox skill, and this electrons will emit, the electron that is emitted from photosystem 2 pass through the electron transport chain to the pigments of photosystem 1. Simultaneously electron in the reaction center of photosystem 1 are also excited when they receive red light of wavelength 700 nanometer and are referred to another acceptor molecule, that is FRS, and then to FD, and these electrons then move downhill, this time to a molecule of energy rich NADP. Here this is the important point, the electron that were moved from photosystem 2 must be replaced. This is achieved by electron available due to splitting of water. The splitting of water is associated with photosystem 2. So within the photosystem 1 water splitting, that is chemiosmosis, cannot occur. So photosystem 2, okay, water is split into H plus oxygen, nascent oxygen and electrons. This creates oxygen, one of the net product of the photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. So this is the oxidation of water reaction. For oxidation of water, 2 water molecule reacts to form 4 H plus and 1 oxygen molecule plus 4 electrons. These electrons will be refilling the electron gap created in the photosystem 2. So we need to emphasize here that the water splitting complex is associated with the photosystem 2, which itself is physically located on the inner side of the membrane of the thylakoid. So it is present in the inner side of the membrane of thylakoid. So addition to these electron reduces NADP plus to NADPH plus H plus proton from stroma. At every step oxidation reduction of electron carrier take place and energy is released, which is utilized in creation of proton gradient from ATP. So the whole scheme of transfer of electron starting from photosystem 2 uphill to the acceptor, down the electron transport chain to photosystem 1, and excitation of electron transfer to another acceptor, and finally downhill to NADP plus is called Z scheme, because it will look like a Z. Like this it will be present from photosystem 2 to electron acceptor and then to photosystem 1, then again excited and electron acceptor, from where NAD pH plus H plus can be formed from NAD plus, NADP plus. Okay, so the number of, so here comes the cyclic photophosphorylation. This cyclic photophosphorylation only works, only PS1, that is photosystem 1 works if it is this cyclic photophosphorylation will not yield much ATP and NADPH. So it occurs only in the stroma lamella. A possible location where this could be happening is in the stroma lamella or thylakoid. So during cyclic electron transport system, the electrons ejected from the reaction center of photosystem 1 return back to its reaction center, that is chlorophyll A. At every step, oxidation and reduction of each electron carrier take place, which releases the energy, and this energy is utilized in creation of proton gradient to form ATP. And in cyclic electron transport system, no oxygen evolution occurs because of photolysis of water is absent. Water photolysis is absent here. So as a result, no oxygen is evolved if cyclic photophosphorylation occurs. Oxygen is evolved only during non-cyclic photophosphorylation. And NADPH plus H plus is not formed in cyclic process. There is formation of only ATP. So here comes a general differentiating table column for cyclic and non-cyclic. In non-cyclic, both photosystem 2 and 1 is present, but in cyclic only photosystem 1 is present. First electron acceptor here is phaeophytin, here is FRS, and here electron expelled from the reaction center is not cycled back because its loss is compensated by electron from photolysis of water. But here photolysis of water cannot occur, and the electron expelled from photosystem 700, that is photosystem 1, is cycled back because photolysis of water cannot occur, and evolution of oxygen does not take place. Here photolysis of water occurs and evolution of oxygen take place. Here NADPH plus H plus is formed from NADP plus. Here NADP is not reduced to NADPH plus H plus. So this completes about the cyclic and non-cyclic photophosphorylation. So this is the first and foremost step in the process of photosynthesis, and it is otherwise called as light reaction. So both cyclic and non-cyclic occurs inside the chloroplast. But the thing is, non-cyclic occurs in the thylakoids, but cyclic occurs in the stroma lamella, because stroma lamella has photosystem 1 only, but this thylakoid has both photosystem 1 and 2. So that is the reason why the non-cyclic photophosphorylation is occurring in the grana or otherwise thylakoids. Okay, so this completes about the differentiation between cyclic and non-cyclic photophosphorylation. Next video we will see about the chemiosmotic hypothesis. Thank you.